We have Dr. Amy Mathers joining us today. She is the Associate Professor of Medicine and Pathology and Associate Director of Clinical Microbiology at the UVA School of Medicine. So thank you for your time today. And also Stacy Helberg and Kristen Lachlan, who are both public health nurses at the Fairfax County Health Department, Division of Epidemiology and Population Health. So we're really happy to have some local health district speakers joining us today. So thank you all, and I will turn it over to Dr. Mathers now.
Great. Thanks, Jenny. Thank you so much for the invite to talk to you today about obviously something very near and dear to my heart. And it's sort of how to think about horizontal gene transfer and how to think about the genomics of that as we get farther along in using genomic tools to understand transmission. How do you think about these little mobile elements that might move between bacteria? And so I will quickly go through it. It'll be a little bit of a whirlwind, and I'm always open to coming back or questions. So I have some conflicts. I'm not going to talk about anything today. Just to set the stage, to orient everybody about where my space is largely, is antibiotic resistance. So this report came out last year showing that the global burden of AMR is the third leading cause of death globally. And a huge chunk of the infections that drive antibiotic resistance, mortality, and morbidity are things that exchange genes of drug resistance. So what I'm going to really focus on, and a really good example of gene of drug resistance exchange, but this applies for ESBLs and for basically any gene of drug resistance that can be exchanged between gram-negative bacteria, which is the Enterobacterialis, so E. coli, Kleb, those kind of bacteria. So ESBLs and CRE, most of the resistance is on plasmids or mobile genetic elements. And again, CRE, we know, have increased over the pandemic. They were on their way down, and now they're back on their way up since 2019. I think, this is true of a lot of things in hospital AMR tracking and nosocomial transmission. They're not really labeled per se. So to spot a CRE, you sort of have to stay on your toe because Enterobacterialis isn't even a name that's given here, right? This is a Serratia, this is an E. coli, and this is a Kleb. And these things are all CRE. So you got to really pay attention because the numbers may be interpreted differently depending on what breakpoints people are using. Some of them may even read as sort of susceptible looking. And so to track these genes of drug resistance, you got to really stay on your toes. But one thing I wanted to highlight, and one reason this may be coming more across your radar, is we are really pushing on the treatment side that clinical labs are doing carbapenemase testing locally because it does inform decision making. And that's because here under this orange bar, these are all the new antibiotics that have come recently to treat multi-drug resistant infections. And it really matters what enzyme you have and how you decide what to treat them with. And so I'm not going to go through this, but I just wanted to tell you that we're really trying to push clinical labs to have some means of carbapenemase testing. And now that there's a ELISA that's about $5 to test the carbapenemases, I personally think clinical labs should be doing this to care for patients. Because it's not all just KPC or CRE. So previously, you could sort of say, well, this is a carbapenemase producer in the state of Virginia. Carbapenemase producing Kleb pneumo is going to be a KPC. Non-carbapenemase producing is just sort of an irritated bacteria that's been exposed to a lot of antibiotics. But unfortunately, we're seeing more and more other types of enzymes that, again, require different treatment strategies. And this is compliments of Shana Bernard and the HAI team that put this together, showing that, we're seeing more NDM. And when you look at E. coli, half of the E. coli in Virginia last year had a new enzyme, a new kid on the block, NDM, which makes it really, really hard to treat. And so the thing is these genes of drug resistance, and I'll get back to this in a little bit more detail in a moment, but, the carbapenemase gene sits on a mobile piece of DNA that can be shared across species. So that's what I'm going to talk about. So this is a case that came to us about a year ago, but it was a pan-resistant Pseudomonas in a urine culture. The patient had actually been discharged and was at rehab, and the rehab facility was calling us and asking us for treatment advice. And we were like, oh, that's really interesting. This patient was just in our hospital and is now somewhere else. But he had gotten a month of healthcare in India before transferring back to Charlottesville. And so we were like, I wonder if that's an NDM producer. And we checked it. And in fact, yes, it was an NDM. So we went back to the unit where he was at in our hospital and did a perirectal sweep of everybody on that unit. And there was one patient that was positive for NDM, but he had 4 species that were positive for NDM. And so Club Quasinpneumonia michigensis, you didn't need to really know these names, but these are all different species of bacteria. So could this be possible that the Pseudomonas transmitted to this other patient? We don't see very many NDMs, so we were like, shoot, I'm not sure what's going on here, but I am concerned about transmission. What about within patient? Did this patient independently acquire all 4 of these species, or was there something going on in that patient's gut? And can we use genomics to try to answer the questions around how this transmission occurred? So you can sort of use genomics. And this is a genomic epi call, so I don't mean to throw too much genomics at you and too much, but most of what we've done to date is short read sequencing. Illumina sequencing is considered short read sequencing. So you take all of the genome, here's represented the nucleic acid of a bacterium, here's the chromosome as a little cartoon, here's a plasmid as a little cartoon. These are not to scale because the chromosome is so much bigger than the plasmid. And you take and you break it into little bits, and then you amplify those bits and read the genomic sequence from each one of those little bits. When you then go to compare a club pneumo to a club pneumo, you have to map it to a reference, and that reference usually only contains the chromosome. And so then you can say, oh, this is the reference, and then our reads look like there's one difference here compared to the reference. And that's how you sort of do comparison of, salmonella or an outbreak of Staph aureus. But the plasmid DNA is largely just kind of thrown in the garbage because you can't make sense of it because it's just too broken up and there may not be a good reference to map to. So to really understand plasmid genomic context, you have to do longer read. And so when you do long read sequencing, which is PacBio or Nanopore, you can then sort of assemble de novo what the plasmid looks like. And then you usually have to do short read to fill it in and cover the gaps. If you don't do long read sequencing and you try to make sense of plasmid, you can really end up in a pickle because you can really mislead yourself. So I'm just going to go through a little bit of some of the stuff that we've done to kind of demonstrate that example and how hard genomics can be, and then I'll finish out with what happened with our case. So I ended up getting into plasmid genomics because of an index case. It was the first case of KPC in the state of Virginia. And it was in August of 2007. This patient had been in Philadelphia and was transferred to our hospital and had a club pneumo and a club oxytoca, both which produced KPC. And so we were able, through a lot of work and a lot of hard work and Southern blots and before genomics, able to recreate this plasmid. And now we've done it with next generation sequencing genomics in collaboration with my mentor at Oxford. And what we found is we had a completely novel plasmid that was carrying KPC and another plasmid. And again, these are the kind of things that get shared between bacteria. So what? Who cares? Except that this is the epidemiologic curve of KPC at UVA. And it looks like an epidemiologic curve of sustained transmission. And what I will say is the difference here is each different color is a different species. So far we've had 31 different species of KPC producers at UVA in patients over time since 2007. And so is it all coming in from somewhere else, or how in the world is this happening? Well, it's happening because the plasmid is moving in between. And my mentor, Derek Crook, helped me do a lot of this work, and he was the head of Public Health England for a lot of this time. It's important to recognize that we do perirectal surveillance at UVA and have since 2009. And so we have a good handle on who gets KPC silently or not, because we were concerned that maybe some of the transmission was occurring because we were not checking silent colonization, and that's where the transmission gaps occurred. So this is our current criteria. Outside facilities, other countries, but as you'll see in that case that I presented, we missed somebody that had come through from India because it's hard to identify people coming from other countries and who need screening. But we try to screen high-risk people, and I can talk more about that if you have questions. And so what we did was we took the first 5 years of our outbreak of those sort of multi-species, and we did whole genome sequencing, mostly Illumina, that short read sequencing, and we randomly selected 17 isolates to do long read sequencing. A lot of this work was led by a bioinformaticist named Anna Shepard. And what we did was we wanted to see, was this a plasmid mediated outbreak? It sure looked like it from both preliminarily molecular work and from some of the other clues that we had. And so again, when I think about genomic epi, this is sort of how I do genomic epi, think about it. so we've got an index case shown here, and what's shown in this line is the same strain. So let's just say this is Kleb pneumo. You have a patient that comes from an outside hospital, has Kleb pneumo on admission, so you know they had it before they got to you, and we'll call that an import. And we define our imports as less than, had it identified before transfer or had it within 48 hours of arrival. And so that's an import. And then, so they're on the ward, let's say ICU X, and another patient comes up with a Kleb pneumo with the same resistance pattern and the same gene of drug resistance, and you go and you sequence it, and it's identical to the imported case. And they were on the ward at the same time. And so that's a transmission. And here on this, we've got the same strain and all these dots representing patients with patient to patient to patient transfer. They're all on the ward at the same time. And so shown here is this really bad hypothetical outbreak of a Kleb pneumo strain, all the same strain. Anybody who does epi work knows that occasionally you'll have a patient that pops up with that strain and wasn't on the ward at the same time as anybody else that had it. And you're like, I don't know how in the world that patient got it. So there are some hollow circles where you're like scratching your head about where the transmission link that you might have missed was. This is what our outbreak looked like. So we had 62 different strains of bacteria carrying KPC, each represented with a different line or dot when there was no other isolate that looked like it. And on that line, what we had is all these hollow circles and big spans of time in between. What I will show is this line here with all these X's. This is all the imports. So this is the Kleb pneumo ST258 that was circulating widely in the United States and in the state of Virginia. So people were bringing that into our hospital, but we weren't seeing transmission of that strain in our hospital. We were seeing all these different strains, all with KPC, not sure how they were transmitted. We also know that, you know, if anybody knows anything about UVA and our epi, there's these huge gaps of time between seeing a strain in one patient and seeing in the next patient. We now know that the hospital environment was involved in this outbreak, and I could talk about that at another time. When we looked for the plasmid to see if it was a single plasmid that was being transmitted between patients, what we found is 66% of the patients had that index plasmid from that patient from Philadelphia, and another 18 had the second index plasmid from his other isolate, the Kleb oxytoca. So the vast majority of this was a plasmid-mediated outbreak. And very quickly, and not to confuse you too much, was we did long-read sequencing, and we found, in fact, PUVA01 carrying KPC was present in a lot of the isolates. So of the 17 randomly selected long-read sequences that we did, 11 of them had PUVA01, so the vast majority. But the problem was is some of those plasmids no longer carried KPC. So it looked like PUVA01 was the vehicle from which things were moving, but it wasn't really straightforward because they weren't even a KPC plasmid anymore. And so what we found was that, again, this is pretty confusing, but KPC was sandwiched here in purple, this red gene is KPC, was sandwiched in another mobile genetic element, and this purple bar in and of itself can move on its own. And so when we really did all the long-read sequencing, we found that we had 11 unique KPC plasmids from 17 isolates in this really hot mess of an outbreak. And so very, very confusing. But what we put together is that it's really complicated the way genes of drug resistance move around. It's a gene inside a transposon, inside another transposon, inside a plasmid that can be shared across a bunch of different bacteria. And so I will say that we saw KPC spreading at all levels, both plasmid transmission, transposon transmission within a cell, and it's a hot mess. And so as I'm short on time, I just want to say that NDM, in terms of its hot mess and move around, runs circles around KPC in terms of mobility. And so right from the very beginning, this is the initial paper that described NDM in New Delhi, or one of the initial papers actually. And you can see there that already right out of the gate, NDM was being seen across many, many different species in Southeast Asia. This is in Dhaka City. And what's also interesting is it too is in a mobile genetic element, and this thing moves around like bonkers. And so when we did our long-read sequencing on our patient, we had the Pseudomonas patient who was on the ward at the same time as that patient who had 4 other species. What we found is there actually wasn't any plausible mode of transmission. The Pseudomonas that we did long-read sequencing on, the NDM was in the chromosome, and that Pseudomonas didn't even contain any plasmids. My suspicion is with that NDM1, he obtained that in India where he had gotten healthcare and didn't transmit it forward, at least that we identified. And for the other patient, I don't know where they got it, but it was on an INKX plasmid that was shared across all those species. And my hypothesis would be in that patient's gut. I don't know which one the patient acquired. And when you're doing this type of genomic epi, figuring out which was the first isolate that they acquired or which one is spreading between patients, it's really important to sort of try to work on the directionality and use genomics to do that. So in conclusion, I'll just say it's really, really complicated, but I do think, especially through pathogen genomics, we're working on trying to develop some tools for plasmid for use for genomic epi. And so hopefully it will get easier, but I don't think the biology is going to get any easier. So there's a lot to learn and a lot to share about this topic. Lots and lots of people worked on this. I'm just the one sort of presenting it. And thanks to the funders, CDC, and I'm just so excited to work with DCLS and VDH on this plasmid project. So I will take questions at the end and hand it over to the next speaker. Thank you so much.